Simplified cell-based overview of coagulation. The cell-based model of coagulation is a more modern understanding of blood clotting that emphasizes the roles of different cell types and their surfaces in the process, unlike the older cascade model, which focused on a linear series of protein interactions. The cell-based model describes coagulation as occurring in three overlapping stages: initiation, amplification, and propagation, with each stage occurring on specific cell surfaces within blood. First, the cell surfaces involved. The platelet, or thrombocyte, is a small blood cell that circulates in an inactive state until needed. Unique properties of the platelet include the alpha and dense granules. These contain many of the factors involved in clotting, such as factor 1, fibrinogen, and as well as factors 5, 8, calcium, and von Willebrand factor. The granules also contain substances to help recruit other platelets to a site of injury, and they also contain substances that limit or inhibit clotting activity. Surface receptors on the platelet are key to platelet adherence and aggregation, or its ability to stick and clump. Next is the tissue factor-bearing cell, or the fibroblast. When tissue cells are damaged, tissue factor is released, and these are going to begin the triggering of plasma factors. And it's on these cells, the fibroblasts, that the clotting process begins. A breakdown of the three stages. At initiation, coagulation begins when blood is exposed to tissue factor from the fibroblast, often at the site of an injury. This will trigger the activation of factor 7 from the plasma, and this initial activity will spur the release of von Willebrand factor from the platelet as well, which will bind to one of the unique surface receptors, helping to anchor it to the site of injury. The tissue factor 7A complex then helps to activate factors 10 and 5 respectively, forming the extrinsic tenase complex. And it's this complex that leads to the production of a small amount of thrombin. This initial thrombin generation is not yet strong because factor 10 is easily inactivated by tissue factor pathway inhibitor, particularly as it moves away from the center of activity. However, this thrombin generation is critical for activating the next phase. The next phase is amplification. The small amount of thrombin generated binds to the surface of the platelet, activating it and other clotting factors, such as factor 5 and factor 8. This activation creates a positive feedback loop, further amplifying the production of factor 10 and its cofactor 5, and a rapid increase in the rate of thrombin generation. At propagation, clotting factors assemble on surfaces of the activated platelets and fibroblasts. Thrombin further activates factor 7A, reinforcing the extrinsic pathway that led to its initial generation. Thrombin also activates factor 11, and factor 9 is activated by both thrombin and factor 11. Factor 9 joins its cofactor 8 to create the intrinsic tenase complex. And it's this powerful complex that reinforces the activation of factor 10 and its cofactor 5, creating a platform for the massive generation of thrombin, or the thrombin burst. Further substances are released from the platelet. In addition to von Willebrand factor to contribute to adherence, ADP and thromboxane help to further recruit more platelets to the area to clump and aggregate. And it is this high concentration of thrombin that is crucial for converting fibrinogen strands from within the platelet into fibrin strands, which cross-link to form a stable and secure clot. As compared to the cascade model, the cell-based model better explains localization, how the clot occurs at the site of injury, and this is due to that initial and important contribution of tissue factor from the fibroblast, which generates the initial process. The cell-based model also better explains individual clotting factor deficiencies. It's easy to see why factor VII deficiencies are so severe when we understand how coagulation is not a linear cascade in which sections of it happen independently, but rather a series of overlapping stages that are influenced by cell-to-cell interactions. Factor XI deficiencies aren't often associated with significant bleeding. It isn't the only trigger for factor IX activation. And it's clear why factor IX and VIII deficiencies, as seen in hemophilia, result in such serious bleeding disorders when we can visualize their importance in the feedback loop that reinforces factor X and the generation of the thrombin burst. The cell-based model provides a more accurate representation of how hemostasis occurs in the body, and it highlights the importance of cellular surfaces in providing the necessary environment for these clotting reactions to occur efficiently.